Fine. So as I said, once the operator got mutated, we generally call as constitutive expression. Why? Because when the operator got mutated, even though regulatory gene synthesized the repressor never go and attaches to the operator. Now operator become free. Once the operator become free, RNA polymerase crosses it successfully and synthesize the three genes in a constitutive manner. Now, when you look into the regulatory gene got mutated, what will happen here? Repressor cannot synthesize. Even though repressor can synthesize, repressor is unable to block the operator. Then again, operator will become free only. So remember, whenever the regulatory gene got mutated, there are two possibilities are there. Repressor protein cannot be synthesized. Even though repressor protein synthesized, most of the time it cannot attach us to the operator. But mostly repressor cannot be synthesized only. All right. So I told you to answer this question in the tomorrow class. If anyone answers, we will discuss that. Okay, tried it. I have given one question. Remember it now. Try to tomorrow without fail this question. I have told you to solve it. Don't forget. All right. So now, if you very quickly recall, what is the differences between the Lac Operon as well as the tryptophan operon? Lac Operon is what kind of a operon we studied, which is called as the inducible operon. But if you look into the tryptophan operon, what I told you, repressible operon. What is the differences between the inducible and repressor? If the inducible operon will be turned on, if there is a presence of inducible. Repressible operon will work whenever there is a presence of repressor and co-repressor. Okay, we'll discuss that. We'll discuss that elaboratively. Okay, now if you look into the tryptophan operon, I have very clearly told you, Lac Operon is also regulated by two type of regulation. What is that? Negative gene regulation and positive gene regulation. What's the difference? How the negative gene regulation can be regulated by in the Lac Operon? Very good. Operator and repressor. Correct na? In the Lac Operon. In the tryptophan operon also same. By the operator and repressor. Positive gene regulation is regulated by the capu protein. In the Lac Operon. In the tryptophan operon, it is regulated by what? Attenuation mechanism. Okay, what mechanism it is? Attenuation mechanism. No point of getting confused here. Okay, now everyone just try to understand. Let me quickly explain you. If you everyone recall, in case of a Lac Operon, there are how many structural genes are there? Lac Z, Lac Y, as well as the Lac A. Now, when you look into the tryptophan operon, how many are there total? 5. So I already told you, you people no need to remember these 5 names and you don't need to waste the time. There are 5 structural enzymes are there. In the last class also, I told you what is the major precursor that is involved here. Remember everyone, charismate. I don't want you to remember the name of the 5 enzymes, but remember a precursor molecule that is required for the biosynthesis of the tryptophan. What do we call it as the charismate? Precursor means starting material for the synthesis of the tryptophan. Now this charismate can be used by these 5 enzymes and finally they're gonna give rise to the tryptophan. Simple. Now let's understand the regulation very accurately. So it is regulated by 2 mechanisms. Negative gene regulation called as a repressor operator. Positive is called as what I told you, attenuation mechanism. Now you only keep on answering my questions. First understand the concept very correctly. It is just opposite mechanism. First one is repressor or operator interaction. Now tell me, again, if we want to understand the negative gene regulation or if you want to understand the positive gene regulation, what is the key mechanism here? The key mechanism is presence of the tryptophan and presence and absence of the tryptophan. Now you think properly and answer my questions. Last time also I have given you one hint. The hint is in vivo, which means within the body. Tryptophan, what it is? It is an amino acid which is required for the synthesis of the protein, which means tryptophan is getting biosynthesized every single time in the body. Correct na? So tryptophan genes are always active in the body, in vivo. But when I use the word presence of the tryptophan. When I use the word absence of the tryptophan, I am only talking with respect to the medium. Medium means in vitro. Correct? Na? Don't get confused over there. So now you try to understand word. The word says that tryptophan is absent. Where it is absent? Medium. Medium. So which means I am a bacteria. When you have not provided me any tryptophan externally, what I will do? I keep synthesizing tryptophan in my body. Correct? Na? If I want to synthesize the tryptophan in my body continuously, does the operon should be turned on or off? Obviously on. So what's the condition here? Tryptophan is absent. Which will become active? Sorry, which will become inactive? Repressor become inactive. So operon will be turned on. Now you all people has to get a valid doubt. Why the repressor become inactive? Look into the operon correctly. There is a presence of 5 structural genes. Operator, where the repressor comes and attaches. Promoter, where RNA polymerase comes and attaches. This is the regulatory gene. Undergo the transcription, translation, produced the protein, which is called repressor. Originally, now the repressor has to come and attaches to the operator. Why it happened? Why it not attaches to the operator? The reason is that even though repressor becomes synthesized, it is inactive. What is the reason why it is inactive? The reason behind that, it requires the presence of a, whenever there is a presence of the co-repressor, it will become inactive. Sorry, it will become active. Now, who is the co-repressor? The tryptophan, which is coming externally, that itself act like a co-repressor. But what is the condition? Tryptophan is absent. Where? In the medium. So now the repressor, even though it got synthesized, it will become inactive. Then what happens? Repressor cannot attach to the operator. Operator become free. RNA polymerase crosses it successfully. The 5 structural genes synthesizes. Tryptophan synthesis will occur continuously. No confusion. Now, tryptophan is present. Where? In the medium. So that tryptophan will directly come and attach us to where? Repressor. Because so far repressor is inactive, which is already synthesized before. What happened then? The inactive repressor become active. Active repressor will go and block what? Operator. Then RNA polymerase cannot cross the operator. Hence, no RNA got synthesized. Then the no tryptophan is synthesized, which means the bacteria starts using the tryptophan which we provided externally in the medium. This is a very simple negative gene regulation. This is not that much difficult. What is more important? Now slowly think and answer my questions very correctly. The positive gene regulation called as a attenuation. First of all, answer my question slowly. Let's consider a condition. Initially, tryptophan is absent in the medium. What happens in the body? Tryptophan mRNA is getting synthesized. What you have done, you have provided me the tryptophan all of a sudden. What I will do? Undoubtedly, I start using the tryptophan which you have provided. Then what should I do inside? Inside also happening before. So what I have to do? I have to stop. Now my question is super clear. I have to stop the entire mRNA, or I have to stop the tryptophan synthesis, which is, see, 50% is completed, example. Then can I stop in the middle, or I have to stop entirely? Suppose you understand, if I stop in the middle, is there any use of that mRNA? Correct or not? So I have to stop completely. So that is exact question. We are stopping in the middle, or we are stopping the entire mRNA synthesis? We have to stop the entire mRNA synthesis. Now you can ask me a question. Sir, 50% is already synthesized. What happens to it? Does it give rise to the protein? No. It is a truncated mRNA, abrupted mRNA. It will get degraded automatically. Correct or not? It is not like that you already synthesized 50%. It's not like that you have to stop only remaining 50%. That is wrong. You have to stop the entire mRNA. Now that mechanism is called as attenuation. So termination of initiated transcripts. This is also completely depends on a sequence called as a leader sequence. This is the one which generally do the positive regulation. Understand the concept very correctly. This is the operon having 5 structural genes. And what is this? Operator. This is the promoter. Anyway, we know very well here there is a presence of which gene? Regulatory gene. No point, no confusion. Can you observe very clearly between the operator and the structural gene E? There is a other sequence. What do we call? Leader. We call it L sequence. If you observe the L sequence correctly, only 10 to 20% of the leader region only going to give rise to polypeptide. Remaining region, 80 to 90%, it is not at all going to code for any polypeptide. It is acting like what? Attenuator. Don't confuse. Attenuator generally act like what? Regulatory region. No point of confusion at all. And this attenuator, exact question, contain 4 sequences. Sequence 1, sequence 2, sequence 3, sequence 4. All the 4 regions of the attenuator are complementary with each other. What does it mean? Which means 1 can pair with 2, 2 can pair with 3, 3 can pair with you understand that? You have to remember that compulsory. Okay. So only remember majorly when 3 and 4 pair with each other, it gives rise to a signal called as termination signal. When 1 and 2 pairs with each other, it can give rise to a signal called as a passing signal. 2 and 3, it gives rise to an anti-passing signal. It never stops. It will continue. The passing means we are stopping it. Anti-passing means we are allowing it. You understand? Termination means we stop the complete process. Now, here comes the question. When 1 and 2 will pair, when 2 and 3 will pair, when 3 and 4 will pair, this is completely depends on the presence of the level of tryptophan. So tryptophan is the major parameter here. Okay, now I'll explain once again. Just try to understand these points very correctly. Use the brain. If there is a, understand, first listen the question correctly. If there is a plenty of tryptophan, is there any necessity for the ribosome to wait to bring the tRNA that is carrying the tryptophan? I am asking a question. There is a plenty of the tryptophan. Is there any necessity for the ribosome to wait for the tryptophan tRNA to come? Why? There's no necessity because tryptophan is already present. Correct or not? So hence, what will generally happen? The ribosome immediately translate the entire leader polypeptide. I told you, right? Leader sequence can undergo the leader can convert it into the polypeptide. In this suppose example, understand, I'll re-explain 2 to 3 times. Suppose leader contain 10 to 20 amino acid. Let's consider. In this 10 to 20 amino acid, which is the leader polypeptide, it also contain 1 to 2 tryptophans, tryptophan codons. Suppose if the tryptophan is abundantly found, the tryptophan, the ribosome never wait for the tryptophan tRNA to come. It's not required because it is already using which is present. I hope you're getting my point correctly. Then what happened? Leader polypeptide can synthesize all of a sudden. I mean to say, it occur very fastly. Hence, you tell me what is going to happen. 3 and 4 will pair. Termination will occur. Correct or not? Now you tell me if there is only very little tryptophan is present, then what happened? Ribosome has to wait some time to bring the tRNA that is carrying the tryptophan. Correct na? Now you tell me at that particular time, do you think leader polypeptide can occur fast? No. So can I say that I have to stop the transcription for some time? Correct or not? Hence 2 and 3 will pay. Which will give, I'll tell you, which will cause the anti-passing signal. I'll explain you with a simple animation, then you will get a brilliant idea. So it's all about the always follow the foreign animations. It will explain very simply. This is also good. Now, first listen.
Necessary to synthesize amino acids if they are not present in the nutrient medium. If the amino acid is present, then the operon is repressed and biosynthetic enzymes are not produced. One example of repressible regulation in E. coli is the trip operon. Two regulatory mechanisms are involved in the control of the trip operon. We will focus on only one of them, attenuation. The trip operon consists of regulatory and structural genes. The promoter
So you can see here 5 structural genes, tryptophan operator, promoter, and can you see that? Leader. Where the leader is located exactly between the operator and structural gene, which is called E.
The region of the trip operon regulates the level of transcription. 5 structural genes labeled trip E through trip A are located downstream of the promoter region. The structural genes are transcribed as one polycistronic mRNA.
Because it's a prokaryote.
Polypeptides.
So what is the precursor required? Chorismate. First, chorismate will activate all these 5. By using chorismate, these 5 give rise to the tryptophan.
Translated from this mRNA are responsible for tryptophan biosynthesis. Between the promoter operator region and the structural genes is the leader region, TRPL. This region is transcribed into an mRNA called the leader mRNA. The leader mRNA.
So this is what? Leader mRNA. That is L region. So L region, it's an mRNA means what are the sequences that it contains? It contains start codon, it contains stop codon, and remaining region called as coding. Very good. Structural gene or the coding region. So coding region means it is going to code for what? Amino acid. Can you observe the picture here? There are in their coding region only what are present? 2 tryptophan operon, 2 tryptophan amino acids are also present. Where? In the trip L region. And if you observe the trip L, if you consider it as 100%, how much percent of the trip L is going to code for the protein? Only 10 to 20%. This complete is trip L. Remaining trip L is basically called as attenuator. That is basically blocking or controlling the tryptophan operon. Now understand.
Contains the attenuator consisting of 4 separate regions.
Can you see this? All of you look into this from AUG to UGA. That is only triple. Very small stretch. Remaining can acting like attenuator. Attenuator contain how many sequences? 4. 1, 2, 3, 4. That one region is located near where? Can you see that? Where it's located? Near the tryptophan codons, which are present on the mRNA of the trip L. Understanding the points correctly. So 1, 2, 3, and 4.
That can fold and form 3 different stem loop secondary structures.
I want you to understand this. This is called leader peptide coding region. Very small stretch. Remaining all called as attenuator. In that attenuator, 1, 2, 3, 4 sequences are present. And that at 4 attenuator regions, out of 4, 1 is present where? In the leader peptide only, which contain 2 tryptophan codons.
Structures by complementary base pairing. Pairing of regions 1 and 2 results in transcription pause signal.
1 and 2. They can form a hairpin loop like structure. This is the exact question asked in 2015. Okay. And 17 also. What kind of structures can form? 1 and 2 will form pass.
Pairing of regions 2 and 3.
Anti-termination. We can call anti-passing. Okay. Anti-termination means it never allow the tryptophan operon to get tryptophan leader region not to synthesize completely. Why? You tell me why? Why anti-termination is happening? Because the tryptophan level is low. When the tryptophan level is low, we have to wait for what? Tryptophan tRNA to come. Then only polypeptide synthesis can happen. When the termination signal happen, when the tryptophan level is extremely higher, there is no point of waiting for the tryptophan tRNA to come because we already having tryptophan. You're getting my point. Then why do we use this two tryptophan not required? So 3 and 4 will
An
perform termination.
anti-termination signal that
So you'll get
allows
it.
transcription to continue. Pairing of regions 3 and 4 results in a termination signal which stops further transcription. Because there is no nuclear membrane in bacteria, transcription and translation are
Or
coupled.
obviously coupled in the bacteria. What does it mean? When mRNA is synthesized, obviously ribosome attaches and start translating the protein as well.
Translation of the trip-L mRNA can proceed while RNA polymerase is still transcribing.
Correct. Now, what is this L region? Trip-L. So, whenever the trip-L synthesized, automatically what happened? Translation is also starting. Who will start the translation? Ribosome. Now, when the ribosome will wait, when there is no tryptophan. It never wait when there is a tryptophan. Now, entire operon is under the control of level of tryptophan. Now you see
The
that.
attenuator mRNA. The position of the ribosome along the leader mRNA plays an important role in the regulation of transcription. After RNA polymerase has transcribed region 2, regions 1 and 2 can pair to form a stem loop structure.
Correct. Now, when 2 and 3 are pairing, anti-termination to stop the termination because the level of the tryptophan is little lower. See that.
This structure causes RNA
Yeah, we will understand
polymerase
that.
to pause and allows the ribosome to catch up to it. Subsequent transcription is tightly coupled with translation. Now regulation by attenuation can occur. If the cell is starved for tryptophan, then the amount of trip-tRNA drops dramatically. In the leader mRNA, there are 2 trip codons in tandem. When the ribosome reaches these codons, it stalls because the specified amino acid is in short supply and the leader peptide cannot be completed. Since the ribosome covers region 1 of the attenuator,
Why it stopped here? Because tryptophan is not there. So it has to wait someone to bring that tryptophan tRNA. Then only anticodon and codon will pair. Then only ribosome can move. So conclusion is very clear. When the tryptophan level is low, what happened? Ribosome will stall. Where? At the trip codon. Why? It is waiting for a tryptophan tRNA to come. Then only it can finish. But when we have a plenty of tryptophan, it never ever stops. Now it halted, it never stopped. It complete the leader polypeptide. How it will form? With the help of 3 and 4. Now see that.
region 2 will pair with region 3. This pair
Form what signal? Anti-termination signal. Why? Because what happened? Ribosome has to wait until until the tRNA comes here. Immediately what happens? Until the tRNA come to this particular region, the entire leader polypeptide synthesis will stop.
pairing is an anti-termination signal, thereby allowing RNA polymerase to proceed, and translation follows to synthesize enzymes necessary to produce more tryptophan.
Where it is happening? You understand the point. Whenever the ribosome stall, what happened? Ribosome is moving, RNA polymerase is moving continuously, and what it is doing? It is synthesizing the tryptophan genes because tryptophan is not there. So it has to synthesize the tryptophan on its own. So when I already told you tryptophan is low, where it is low? In the medium. Hence inside what has to happen? Tryptophan has to synthesize. Now can I say that RNA polymerase continuously synthesizing the 5 structural genes, but ribosome is stalled. Correct now, which means leader polypeptide is not getting synthesized. Now you see the opposite.
If the cell is not starved for tryptophan, there is enough trip-tRNA.
So enough trip-tRNA is present. Then you tell me what is going to happen.
When the ribosome
Does this will happen? No. So what kind of signal will form now? 3 and 4 will form. They will form the termination. Termination of what? Trip mRNA. Then which polypeptide can be synthesized? Trip L polypeptide can be synthesized.
So reaches the 2 trip codons, 2 trips are added to the leader peptide. The ribosome then continues to the stop codon at the end.
Where it will stop? In the stop codon. So which is getting synthesized? Trip L is getting synthesized. Inside what will happen? It will stop.
1 of the leader peptide. This prevents region 2 from pairing with 3. Instead, region 3
3
pairs
pair with 4. What is stopping now?
termination signal for RNA
Trip
polymerase.
mRNA stops. So now I will write the conclusion here. You tell me, understand the conclusion. So very simple. The level of the tryptophan is low. Where it is low? Very good. Tryptophan level is low in the medium. Okay. Then medium in the sense I'm writing as the in vitro. Okay. Now you tell me in the in vivo what has to happen. In the body, what has to happen then? Very good. Tryptophan mRNA has to synthesis. Very clear. Correct, na? If the tryptophan mRNA wants to be synthesized, what has to pair?
3 and 2.
No.
1 and 2.
Very good. 1 and 2 has to pair, which we can call passing signal. Or 2 and 3 also can pair. Ultimately, what they will do, pass or anti-termination signal is stopping something. You're getting my point very correctly. Now you tell me opposite here, tryptophan level is extremely higher. Where? In the medium. Now, is there any necessity to synthesize the tryptophan mRNA? No. What I have to do, I have to stop. What I have to stop? In vivo mRNA synthesis. For that, what has to pair? 3. Then tryptophan mRNA synthesis will stop. Which will continue? The tryptophan which is present in the medium, because we have provided so much of tryptophan. So the conclusion is, you have to understand, if there is a plenty of tryptophan, what is going to pair? Tell me enough. 3 and 4. Correct or not? Yes. If the tryptophan level is low, what has to pair? 1 and 2 or 2 and 3. Simple. Correct na? Now you tell me, can I use one word? When 2 and 3 got paired, can I say that all the tryptophan regulated genes are getting synthesized? Correct or not? Don't get confused. Here 2, that is how you will get the question. Don't get confused. When there is a low tryptophan level, where the tryptophan level is low? In the medium. Correct na? Then tryptophan synthesis has to continue where? In the body. So can I say that all the tryptophan genes are getting expressed?
Yes.
Correct na? That is what I am saying.
Yes.
So my conclusion, when 2 and 3 paired in the in vitro condition, all the tryptophan 5 genes are expressed in the in vivo. Correct or not?
Yes.
Then opposite is what I can tell you. When in the in vitro, what is there? Abundant amount of tryptophan is there. Is there any necessity to continue again tryptophan genes? So what should I do? I have to stop it. So I will pair 3 and 4. Then trip mRNA will be stopped. Understand? Once you see the question, you'll get confused. Okay. So you understand the concept very correctly. Now again, there is a very good question. Now, does the negative and positive gene regulations are happening simultaneously or step by step? Again, all will happen simultaneously, because everything is decided by what? Presence of tryptophan. Correct na? So, if only operator and repressor mechanism is existing, the transcription rate just 8 to 10 folds, that's all. Okay. The rate we will express in the form of a fold. Okay, at what speed, at what accuracy it is happening. But when we add this attenuated mechanism, it is happening 560 to 700 fold faster. Means the accuracy of the operon is much higher when there is a presence of the attenuation mechanism. Okay, I will ask you simple questions, because even though you read 100 times without understanding, there's no use. Okay, only focus on understanding and just tell me. Very simple questions. Negative and positive gene regulation. Okay, answer the questions. Tryptophan, I'm talking about negative gene. Slowly understand. Tryptophan is present. Where in the exam, they never use the word medium. Okay. They never use the word in vitro. They never use the word in vivo. Tryptophan is present. So where it is present? Medium. Then operon will be turned on or off? Undoubtedly turned off. What is the correct only? No, that's why don't get confused. Operon is sorry. Tryptophan is present where? In the medium. See, I am a bacterium. You have provided me the tryptophan in the medium. What I will do? Whatever the tryptophan that is synthesizing in my body, I will stop it. Correct or not? Correct na? Then tell me slowly. What is the status of the repressor? Wonderful. Repressor will be active. Correct or not?
Yes.
When the repressor, whenever repressor become active, then only I can block the operon. Correct or not?
Yes.
Now you answer the question. Whenever in tryptophan is absent, where it is absent? In the medium. I am a bacteria. You are not providing me anything externally. What should I do then? I have to synthesize something in my body. Then the operon will be turned on. If the operon wants to turn on, repressor should be active or inactive. No confusion at all. This is what negative gene regulation. Now let's come to the positive. Okay. Now, again, you have to consider presence and absence. Slowly think. There is a presence of tryptophan. Where? In the medium. Wonderful. Is there any necessity to synthesize tryptophan RNA again? Undoubtedly, no. So what can I say? I have to turn off. Wonderful. Then what has to pair? Wonderful. 3 and 4 has to pair. Termination signal will occur. What is getting terminated? Tryptophan RNA is getting terminated. Correct or not? What is continuing? Trip L is continuing. Wonderful. Okay. Now tryptophan is absent. Where? In the medium. Then, so tryptophan is absent in the medium, which means that I have to synthesize the tryptophan in my body. Correct, na? Then, mostly remember 2 and 3, which we can call the anti-termination. Termination of the tRNA is, tryptophan RNA is not happening. What happens now? Tryptophan RNA synthesizes continues. That's all. Beyond this, there's no way of asking the difficult question. But that statements makes you confused. Okay, you have to be very clear with it, because by tomorrow, if I ask opposite, everyone will forget. Okay, that's what we have to remember it very accurate, right? So this is a simplest gene regulation in case of prokaryotes. Okay, but in the molecular biology, we have to give super importance to the eukaryotes. Okay, but every time they'll give the question from prokaryotes also on eukaryotes. Now, in order to understand the prokaryotic gene regulation, we read Operon. In the Operon, we read only 2 levels of regulation, negative and positive. Very simple. So you understand a simple prokaryotic organisms are only having a multiple 2 levels of gene regulation. Then imagine the eukaryotes. We are highly complex. Our genome is extremely complex genome. Hence, we have nearly 6 to 7 levels of gene regulation. See, very simple point. What exactly the word gene regulation means? I don't want this protein to be synthesized. Why? Because if this protein start functioning, it creates greater level of mutations. So I have to stop this protein not to get synthesized. Now you tell me from where I can block this synthesis of protein. Can I tell I can start the blocking the synthesis of protein from the DNA level itself? Correct, na? I never allow the DNA to synthesize mRNA. So I can block there. So mRNA got synthesized. Can I block the mRNA even in the processing level, post-transcriptional level? That also escaped. I can degrade the mRNA. That also escaped. Protein got synthesized. I can block the protein synthesis also by using any antibiotics. Protein also synthesized. But I hope you remember, whenever the protein got synthesized, what do we call that protein? Naive protein. Correct, na? So I can block the protein at the post-translational level, because naive converted into native with the help of post-translation. I can block there also. If it skipped, protein synthesized where in the cytosol. But if it want to show its action, it has to reach us to the targeted area. So I can block the transport. Even though it reached the area of its target, still there also I can degrade. My ultimate goal is I don't want to allow this protein not to become functional. So I can regulate at any level. Okay, so this is what exactly we have to read the eukaryotic gene regulation. So now first we can regulate from where? DNA. It's so simple, you have to answer with normal common sense. Okay. DNA, I'll explain the simple way. Generally exhibit in heterochromatin and euchromatin. Correct now? I without diagrams only you can tell easily, but it's tough. This is the DNA. Where the promoters are present on the DNA. Correct now? Now you tell me how the DNA look like. Is it in a relaxed form or is it in a condensed form? Is it a condensed form? I'll send all to 10th standard. DNA, whenever it is in the condensed form, this is how it look like. Correct na? Of course, chrome. See, okay, let me draw the DNA. This is DNA. This is in condensed form?
No.
No. Forget about DNA. I'm talking about the gene. Okay. This is the gene. Now, is it in condensed form? Okay. This is gene. Is it in condensed or decondensed? It is in the condensed form. Correct na? You can understand the arrangement itself. Now, this is the gene. There is a promoter here. Can I say RNA polymerase enzyme can easily recognize the promoter? Hence, the transcription of mRNA occurs easily. Translation also occurs. Now, when the gene expression is happening, see, whenever DNA is in the decondensed form. Decondensed form means relaxed form, also called as euchromatin. Correct na? Euchromatin means gene expression. Correct na? Hence, whatever the protein, I don't want to allow it, but we are allowing it. So now I want to control the gene expression. I don't want the protein to be synthesized. So I have to convert the DNA from euchromatin to the heterochromatin. So somewhere promoter is located, somewhere hiding it is. RNA polymerase can recognize that? No. Hence, so there is no protein synthesis. Now the question is, no one can convert the DNA, never convert heterochromatin and euchromatin on its own. It is not possible, right? So someone has to convert the DNA into euchromatin and heterochromatin. Who will do that? Histone proteins. Correct na? What are histones? Basic protein. DNA is acidic. Correct na? So basic protein can easily wrap around the DNA and make the DNA become inactive. Right? By that it will be decondensed. We'll understand the mechanisms in the next class. Okay, very elaborative. Okay, by day after tomorrow, molecular biology will be done. Okay, we'll start the toughest unit, unit 13. Okay, the techniques. So from Monday, I'll start the plant physiology wherever we left over. Just one topic only we are left with. Fine, huh? And leave.